Hello, everyone. I hope that you have started a very excellent day today and happy Monday. Today, we want to introduce Professor Brooks. Um, and beforehand, I would like to give you an introduction about myself. I'm Behnaz Akbari, second year PhD student in chemistry, and I will be moderating this event hosted by Purdue University student chapter of Nord for Rare. It is my pleasure to introduce our distinguished speaker, Professor Brooks from Department of Genetics, Genomics, and Cancer Science, University of Leicester, who brings a wealth of expertise spanning multiple disciplines. Professor Brooks' career journey is truly inspiring, starting from a background, mathematics, physics, and engineering. He later incorporated chemistry to transition into the field of medicine. He then pursued a PhD in medical genetics technology, which led him into groundbreaking research in disease genomics and bioinformatics. Over the years, Professor Brooks has made significant contribution to the field, serving as a professor of genomics at the prestigious Karolinska Institute in Sweden before taking on his current role as a professor of health data science in the UK. His expertise in genomics, medical technology, and data science continues to drive innovation in healthcare and biomedical research. Today, we are honored to have him share his insights with us. Please join me in warmly welcoming Professor Brooks. Professor Brooks, please start.
Thank you. Thank you for the invitation, Benes. And I hope what I can present today is of some interest to you. Thank you for attending the talk. I've got quite a few slides, but I'll fly through them. I think with talks like this, you need to go away with some general concepts, not lots of specific details. So I'll try to skirt over the details, but of course, if you want to interrupt at any stage or ask for any further details, just do so. Let me share my screen then and see if this works. Is this working? Yes?
Yes,
Good.
it works.
Looks good to me.
Yep,
Yes.
that's awesome.
Okay, so I've given it this title. Oh, thanks for the balloons, Robert. I've given it this title, which I will elaborate on, because I want you to be clear about what I'm telling you today and why. So the title is Research Asset Discovery in Large European Projects or Networks. What do I mean by a research asset? I mean anything that could be useful in research. This can be materials and all sorts of information, from metadata, data, results, knowledge, and so on. So anything that might be useful to find to help your research. What do I mean by large European network? Well, the tools, the approaches I'm going to present today have been used in many projects, past and current, and some we're applying for funding for the future. Perhaps the most important, though, is this idea of, oh, and I should say in here, I've messed up. I was going to tell you about two projects, EDIRA and EPND, which is why I put them in red. I'm actually only going to do EPND to save a bit of time. So I'll tell you a bit more about EPND at the end. But most importantly is this idea of what do I mean by discovery? Now, in some aspects of bioscience, discovery is about finding drugs. I'm not talking about that. I'm talking about finding assets. So it's very important to understand, especially when you're talking about data, that discovery is distinct from sharing, is distinct from analysis. However, if you do any of those three things the wrong way, you end up doing the other two, if that makes sense. So it's very important when you're setting up systems for people to discover an asset that you don't inadvertently let them get to do some form of analysis on that data, or indirectly then to get their hands on the data. And this is the challenge with discovery. Also, it's important to realize discovery is not the same as analysis in that we're enriching, we're not purifying. What do I mean by that? With a discovery approach, you want to give someone a good sense of what exists. You'll have a bit of false positives, probably a lot of false negatives, but you won't have exactly and only the things they're interested in, because that's getting too close to precise analysis. So it's a difficult balance one has to strike. Okay, so the talk I'll split into 4 sections, a bit more on discovery, some of the standards you might need to do this, an example of some software that's available to you if you want it, and then 1 example project, not the 2 I initially intended. So discovery. This is the way you think about it, I guess. There's a discovery step, which is often based on some kind of metadata or some kind of safe querying of data, by which you can discover a thing exists that you're interested in. Then there's some process that happens by which you request the asset, and then if you're approved, you can get on and get your hands on it and analyze it. That's the way we think about it. You can think of that like eBay, right? You go on an eBay site, you find the thing you want, you discover it, and then you make a request for it. You usually have to pay with eBay, and then it'll get posted to you. But there's an extra step here, which is often overlooked, and it's a very important step. It's this interrogation step. Discovering something in research is generally not sufficient to know that it's exactly what you need. You need to dig deeper to be sure that you want to put the time and money and effort into requesting and accessing that thing. You don't want to get your hands on a data set after 6-12 months of ethics approvals to find out it wasn't exactly what you thought it was. But you can't give that much detail about the asset at the very first discovery stage, because that's really an analysis process. So this interrogation step is very important. There's various ways to do that. You could just give someone an email address so they can start asking questions by email, but you can also support this interrogation step with various technologies. But that's the flow of discovery. Some people say you should set these things up in a centralized system, some in a federated system. I think it's a silly argument, because really you can make hybrids of the two. And increasingly now, this is anyway being done on the cloud, which is at the same time both federated and centralized. And you don't have to set up all of your processes with the same architecture. For example, you might have federated storage, but centralized discovery. You might do your curation in a central place and then do the analysis in a federated manner. You see my point, that all these different steps don't have to follow the same architecture. Okay, so then you think, well, okay, we have the data that we want to discover. We have a tool, some software, Caffe Verium, for example. I'll say more about that later. So you've got to expose those data that you want to make discoverable to that software. Big no. That immediately raises lots of risks of accessing the data when you don't intend people to, finding out information about private individuals and so on. So this is really not the way to do it. Instead, what we do is we create or export metadata. That's a description of the data, and some anonymous or safe version of the data. Those are the items that then get exposed to the discovery software, so that there's no direct connection through to the underlying primary sensitive data. So what have I told you about discovery? These kind of facts. It's really about enriching, not purifying for the things that exist. It can exploit different or safer forms of the data or the metadata. It's really not necessary usually to connect to the underlying data. Then it must proceed, but in a very important interrogation or negotiation step to make it work. You don't have to use the same architecture for your discovery as the storage, sharing, or analysis. You can employ hierarchical networks. I haven't said anything about this, but you can imagine networks within networks and hubs for each of those sub-networks. And obviously, to make all of these different systems work, people have got to agree on how those different systems are going to interoperate. That means standards. They have to define and follow some standards. So a few more slides now on standards. There's lots of standards you could imagine relate to this space. I've listed some of the main ones here. I'm going to say a bit more about two of those because these are two where I co-lead some international efforts to develop these standards. That's DUC-CCE and the Beacon standard. Okay, DUC, first of all, DUC-CCE. It's actually two standards. DUC is digital use conditions, CCE is common conditions of use elements. They're designed to work together, but you don't have to use them both together. You could use DUC alone or CCE alone. Why do we need standards in this space? Well, it's pretty obvious. Use conditions, if you don't understand what the conditions of use are, we can't go sharing and reusing things. But at the moment, there's a real mess around this. And so people just don't know which way to go when it comes to trying to enable additional sharing and use of assets. So that's why we need a standard computer readable way to represent data use consents, permissions, and conditions. So we've come up with this thing. It's taken us a good few years. It's now published in the summer of last year, so you can get the details from publications. The DUC part is the data structure. That's why I've drawn bright white lines. The CCE part, that's the taxonomy. That's the concepts within that structure. That's why I've drawn white lines around the boxes here. And obviously you could use the structure with some other taxonomy. So you could use DUC alone. You could use the taxonomy with some other structure. Hence you can use CCE alone, but they work together. So if I give you an example, if I elaborate more on the DUC part here, the structure is as follows. There's a placeholder for the concept of use, a placeholder for the permission rule. So can you, can't you, must you. The scope, how widely it applies, and then any other details you want to give. An example would be the concept of use by a company. Then the rule would be it is allowed. And then it's allowed for all of the samples in the biobank. But some details are only once the results are published. So you can see how that structure follows and can be applied to pretty much anything. The CCEs, we've spent 2 years testing out and distilling out what would be purely atomic, non-directional, generally relevant CCEs, concepts of use, and here they are. We've now got 35. These 20 or so are published. We're still testing out the extra ones. So a couple more examples on the end there, on the left there. So condition, regulatory jurisdiction exists, and you've got to work within that for the whole of the resource. Or second one, use for profit motivated activities, that is permitted, but only for some of the samples or some of the resource. What about re-identification of individuals? That's a use that is forbidden for the whole of the resource. So you can see how generally flexible this is once you've got those preset set of concepts. And on the right just shows we have lots of software. You can go online and find this now and start playing with it. There's tutorials, there's help text and documentation there. Please feel free to play with it. If you a set of use conditions is what we call a profile. You can make your profiles here to play with it. You can export those profiles and you don't have to leave them on the site. You can leave them on the site if you want to, or you can upload them later if you want to develop them further. We're looking at how AI can interplay with these profiles, and it's very, very powerful. But I won't say more about that today. So feel free to go and play with that. 2 papers out last summer, as I said, and lots of early adopters already. Second standard, Beacon. This is a product from the Global Alliance for Genomics and Health, a global standards body. It is an API. It is a way for computers to talk to each other to execute discovery. It's led by myself with Geordie and Michael on the left, as you see. An example, have you in your data set seen any deletions in this region on chromosome 9 in glioblastomas from patients in a data set, and so on, with unrestricted access? So you can get a sense of the kind of queries that Beacon would let you express. And then you essentially tell one computer to ask all the other computers, do you have anything that would meet those filters? So Beacon in a bit more depth, as I say, it's a J4GH standard. It's actually not a full specification. It's a framework, it's a concept, it's a way of doing things. When you implement it, you have to use certain models. There's models available, but you could also apply your own data models. We refer to lighting a beacon, which simply means setting up a beacon service. And most critically, it's the implementers that decide what's made query, where from, in what networks they respond to, and the like. Now so essentially Beacon allows the system to ask one computer to ask a question to others, and for those others to respond. What it does not do, it doesn't take care of security, IT security, data privacy, anything else like that. Those are additional challenges, and one API can't solve everything. So Beacon sticks purely to the challenge of expressing and responding to queries, discovery queries. Here's that in a graphical form. At the bottom there, you see this is the Beacon service. It would have these different types of endpoints, and the user would come in and express a query where they might be asked about what information is available in your service, what endpoints do you have. It might express some filters, some parameters, and so on, say what pagination and what type of response it wants to get. And then the system would respond, giving the relevant information, either as counts, yes, no, data model handoffs, and so on. So very simple design, but the API has to be very precisely defined. And here it is. Here's what a request would look like. The framework in blue is always standard. The bit you adjust is the bit in orange. Here we're saying, I'm interested in discovering any data about EFO. This is an ontology term for a certain cell line. So it's a standard name for that cell line that all computers would recognize. And I only want you to look in your biosamples section of your data. Here's how a response would look. There's a response part here, which is standard. It just says, yes, we found something, 147 of them. But down here, you could also optionally give the IDs of those samples so that someone could then follow on with an access request. So that's a sense of what Beacon is and how we express our use conditions. And you can see this already starts to become a powerful toolkit to enable discovery activities. Okay, so how do you put all of that together with software? Well, those standards and many others are used by various groups to provide software to support discovery. So we've done that over now 15 years. We've been developing this. We're in our third iteration now. It's a very advanced, fully featured software. The idea and our tagline is the software shares the existence rather than the substance of the data. This software sits beside your existing biobanks or databases, so there's no direct connection into your sensitive data. It doesn't require you to replace or alter any of your local solutions. So to save a bit of time, I'll just go through the left-hand side here. It is safe, practical, flexible. You get to control everything. There's nice admin interfaces. It's modular, so you can use the bits you want. Standards-based, and support is guaranteed for 5 years, and it's all open source. Many, many different projects have used it for discovering patients, for discovering biosamples, metadata, data sets, for discovering individuals, researchers, experts, mutations, obviously, drug toxicity, evidence, publications, biomedical images, and so on. So you can see it's flexible in what you point it to, what you make discoverable. And some of the bigger projects are on the bottom there. This is the architectural diagram. Don't worry about the details. Caffe Verion provides query interfaces, login systems, results, admin interfaces, and an underlying query engine that can be fed with all sorts of different types of data on the right. It can be set up in many different ways. So this might be your installation in the middle here, talking to another installation here, and a network of 3 others, so 5 in total. Each of these lines is optional. So Site A might decide it doesn't want to make its content discoverable to Site C. And so you can imagine all of these have peer-to-peer relationships. It's a fully decentralized network. This instance of the software in some site here has also set up a separate network so that he can be, with the same software and the same content, be part of several different discovery networks. If you've got sensitive data, you can put copies behind a firewall so that they never get contacted by the outside world. The outside world talks to the red copy here, and then these blue copies talk to the red copy to get the queries. So that ensures its safety. And if your data are of different types, you might have the patient clinical data in one database, you might have the genomics data in a different database. A query comes in with asking about both the clinical data and the genomics data. So the query gets separated out, those parts, to the different databases. The results come back and get integrated and returned. So you can have all your data distributed. That gives you a flavor of its flexibility. Many, many different query interfaces, as you see, for all the different aspects that you might want to query on. And that's all modular as well. So those can be mixed and matched and adapted as needs be. Controlled semantics are in there if you want to impose public ontologies or your own vocabularies and so on, you can. Very powerful feature in here, which we've developed and people are very interested in, is the idea of similarity searching. So you're not finding a thing, you're finding something like your thing, similar to. What does that mean? Well, here's an example. The two people in the middle are identical. They both have difficulty walking and have macular degeneration. So they're identical. The one on the left, I hope your screen's the same way around as mine, has macular degeneration. So they're similar to the two in the middle, but they're not the same because they don't have difficulty walking. The one on the right has shuffling gait. So they only match one of those traits, but it's not even a perfect match. It's shuffling gait as opposed to difficulty walking. You start to get a sense of similarity can be powerful, but it can be complicated. So the way we handle this is what's called graph databases, essentially all the different phenotypes you could imagine in a HPO, from the HPO human phenotype ontology are put into a graph. And then we pre-calculate all the relationships between all of those different ontology terms. That gives us over 3 million relationships, which are pre-computed with a number on them. And these are only the relationships. There's a mathematical calculation called REL, only the things that are above 0.1. It goes from 0 to 1. If you went below 1, the numbers would increase exponentially. But you don't really want to find things that have very, very, very minimal relationships anyway. So this has been done, this is available, and this is now going to be made available very soon as an online service. So you'll just be able to tap into that via an API. That's what's on the right, but you can see I've put a human in the middle here, a patient connecting to 4 phenotype terms, right? So you can see on the left, 2 patients can be connected into these phenotype graphs. And so by using the phenotype similarity, you can determine similarity of patients. So you could go in with one patient and it could find another patient that is similar based on the phenotypes they possess and the similarity between those phenotypes. So we build the patients into these graphs to allow those kind of similarity searches amongst patients based on their phenotypes. You can extend that further. For an example here, genetic variants are related to genes, which are related to reactions, which are related to pathways. You can put all of these things into graphs, and this allows you, for example, if you have a database of genetic variants, you can query it by asking about pathways, because all of those relationships are known to the graph. So you say, I'm interested in a certain pathway or a certain chemical reaction. The system knows which genes are involved, and then it looks up in the data for which patients or samples have variants in those genes. You get a sense of the power of this anyway. I think I've put enough time into that. That just gives you a sense of how far we're pushing this software based on standards for discovery in lots of big projects. Lastly, I'll just give you one such project that we're partway through. This is called EPND, European Platform for Neurodegenerative Disease. Leicester here, as you see, is one of the coordinating groups with Gates Ventures. This is Bill Gates' private investment office, and Maastricht University and UCB, the big pharma company. Lots of partners, 20 odd partners in total. We're interested in neurodegeneration. It's a very challenging field, lots of people working on it, lots of places, lots of samples. You need biomarkers. You can't just chop someone's brains out and have a look at it. You need biological markers and digital markers to assess the disease and the progress and the drugs. So we need better biomarkers. That's only going to come if we all start working together. People don't work together, they compete. So by providing this platform, we're giving people a way to make all of their data, all of their samples, all their interests discoverable by others, requestable by others, and to promote sharing and collaboration. So this is just that design. In the middle, you've got the technical hub, all the things it can do, and then flanking that, all the training, education, support services, and so on around that technical hub. Technical hub has a big focus on discovery. That's as I've just explained to you, but also supports data analysis and collaboration, both for data and for samples. Lots of people and groups not in the project are also collaborating with us. As I say, we now have, I think, something like 100 cohorts with data sets and samples listed in here. This couple of quick screenshots, it just basically says, look, it's a discovery interface. You can do filtering, you can find things, not only in a central catalog, but also in a federated manner to lots of those different cohorts that I referred to. And once you've found something of interest, whether it's stored centrally, it's metadata stored centrally, or it's data stored centrally, or whether it's a federated relationship, you can get further details by clicking on it. And then if you're interested, you have the request access option on the right here. That can trigger up either a request or an expression of interest where you can go and do that negotiation process I mentioned earlier. And underlying it all, there's a very powerful engine, a cloud-based environment for safe workspaces. So if you do find a collaborator or you want someone to put the data, there's a safe space to go and put the data and collaborate. That also supports federated learning, federated analysis. Last slide is a list of the relevant, some of the relevant people we work with on all of this. Some major collaborators on the top there. On the right here, previous members of my group that have done so much towards what I presented today, so I'm showing them. And on the left here, the current members of my group. So I hope that was of some interest, and happy to take questions.
Thank you so much. It was fascinating. That's great.
And I stuck to
Okay.
time as well. How's that? It's even better.
Yeah. Thank you very much.
So don't feel
Okay,
obliged to ask
so
questions, but you're welcome to.
Very
yes,
good.
if there
Thank
is,
you.
yeah, if there is any question, uh, you can also put it in Q&A or just open your microphone and ask your questions, please. I have 2 questions for you, but
Who's first?
yes, who is first?
Looks like it's you then.
Go ahead.
Okay. Okay, so I will ask you the questions. Okay, so thank you so much. It was really fantastic because I have also the same as you that have some chemistry background. So I'm very new to the bioinformatic
Okay.
tools, especially for rare disorders. And this is really interesting actually because in chemistry, as you know, it's we heavily think of the as you said, we must focus on the biomarkers, and we think of like how to design the research projects and think of the omics data most of the time. So I wanted to ask you that which maybe I missed that, but which one of these platforms that you introduced we can like upload our omics data
Mhm.
and then expect to check the biomarker similarity between the patients, for example. How we can do that? Is it a possibility of using any of these platforms
If you're
to
talking
give
about genomics markers, or
genomics
multi-omics,
marker? I was
I
thinking
mean,
about omics, to give you more space about
yeah. So
the answer.
the problem with omics is it's in the clues in the name. Omic means very large, right?
Yes,
So
it is very large.
lots and lots
Yes,
of variation, lots of transcripts, lots of whatever omics, metabolites, and so
yes,
on. And
yes.
so similarity between two people on any omics
Mm,
dimension
yes.
is hard to define. You might decide that certain genes are more important in determining similarity or less important, or you might be focusing on haplotypes as opposed to single positions in the genome. You might want to define it in terms of transcripts from pathways rather than individual transcripts, or where they're expressed rather than where or when they're expressed, or how much they're expressed. There's so many different ways you could, depending on your question, define similarity. So that's not something we try to do. That is data analysis, right? What we're saying is, if you've got a very structured domain like an ontology, then it's possible to, from the ontology structure, have a very consistent and well-defined way of determining similarity between any two items in that ontology. The maths behind this, there's different approaches. Some get more and more sophisticated. For example, you can look at some reference data sets, and when the same phenotypes are turning up often in patients, that implies they're similar, right? Rather than just the fact where they are in the ontology tree. So there's different ways you can weight this, put weightings on these similarity measures, but you can only do it in the context of something very structured and defined like an ontology. The idea of doing that for discovery across something, a multi-omics approach isn't realistic. I would say that's about analysis, not discovery.
Because, one of the problems I think in rare disorders in overall is that we don't have such a platform for omics data to give us a sort of direction to analyze our data based on the patient's clinical
Yeah.
you
Well, so
data.
you're right. A project's just finished in Europe. I was
Yeah.
co-coordinating this and leading the IT, called Solve-RD, Solve Rare Disease.
Um,
If a person goes to the doctor and they're lucky enough to get a genome scan or some kind of genetic testing, there's only about a 1 in 3 chance that they'll be diagnosed from that scan. Maybe 40% if you're lucky. So we said, why is that in this project? And there's many reasons why the success rate is so low. But our approach was to find 20,000 patients with their genomes from around Europe that had failed to be diagnosed. Put them all into one place, to redo the processing of the data in a standard way, to apply other omics to it, so long-range sequencing, other transcriptomics, things like this, RNA sequencing, and try to put all of those data together and see if things fell out that wouldn't fall out normally. And we managed to diagnose about another 15% beyond what you'd get in a normal diagnostic lab. But there's still only 15%. That means you've still got maybe a third or more, maybe a half that you struggle with. That reflects the complexity of human biology, I'm afraid. And so you can only go so far with this. But to do that, it was a 5-year project, lots of groups, 20,000 cases had to come together. The idea of having a website somewhere that you can throw your genome in and suddenly, get an answer, I think is we're not there yet. Maybe AI in the future would do this, but I think it's a long way off.
It is it's a dream for me, because I work around the projects that are mostly regarding, lipidomics and metabolomics. So
Okay.
I'm thinking that I'm not a computational chemist to come up with some platforms or ideas about this, but I'm hoping that the platforms like yourself, like how you explain, hopefully will go to that direction in the future.
Well, we're
And
certainly looking at AI, and I said that a bit tongue-in-cheek about omics, but the way things are going, maybe
yes,
some kind of
yes,
AI approach. The trouble with pattern matching is you've got AIs are just pattern matching; they're not intelligent. And the problem with that is you've got to have the reference patterns for them to learn on, to train on. So
yes.
maybe with enough cases we can and enough databases all joined up, we can have a training set that's reliable and big enough. Who knows? I will just a thought come to me. I know because I started off studying medicine. In rare disease, the expert old consultants, a patient walks into their room, they can tell from the face of that patient, rare disease patient, what genes or pathways are probably affected. The young doctors can't do that. They don't have the experience. They don't have a lifetime of experience. But you can take a photo of that individual, you can throw it at an AI machine, and it can make a pretty good prediction of what genes or pathways might be affected to the level of the experienced consultant. So this gives ways for young doctors to, help with their diagnosis. So AI is going to do more and more all the time, I'm sure.
Yeah, or at least I'm thinking that for certain disease, because I always say, I mean, all the researchers, I think they believe this statement that there are some symptoms that
Yeah,
overlapped in many rare disorders.
yeah.
So if we have this similarity, hopefully we can,
So
you
the
know,
problem is the data. The problem is the
the
data.
data.
If if you look at the average data set for a patient, you've probably got 2 or 3 phenotype terms.
Yes, you're
You
right.
need at
Mm,
least 6 to even
at least
begin to
6.
find similar patients. Ideally, you want 20, 30, but the
Yes.
clinicians or the researchers just don't do the work to provide those data. So you're starting with such shallow data per patient that it's very hard to use phenotypes to find those similarities. The genotypes, there's so much diversity in the genotype. If you don't know which gene or pathway you're looking at, you'll always find something similar somewhere in the genome. So the problem is in the data. It's just not good enough yet.
Maybe I think what I can understand from this discussion is if we can educate people and educate patients, I believe, to share their, clinical data to help the
Mm-hmm.
science, I think that would be great. The problem is not patients, I believe. It's all about
Yeah.
um, they are not diagnosed properly at the right time. So that's
Yeah.
why our research also is behind, and we cannot come up with a solution, at the right time.
No, there's many
Um,
aspects to it. Ethics,
yes,
governance, data security, consent, time of the clinicians to do all of this. There is a project you might look up called Patients Like Me, where the patient can go to their doctor, get their clinical notes, get all their genome profiles, whatever, and upload them to this site and then make them available for anyone in the world to access.
that's excellent.
So
Mm,
that's
yeah.
one way. And someone's been running that for 10, 15 years. That's been running now.
And I promise this would be the last question I'm asking
Okay.
you, but that's like so important, I believe. Uh, uh, but please interrupt me if you have any questions. Something. Yes, Juliet, please move on.
No, first of all, it was a great presentation, and I'm sorry the link didn't work, so we couldn't have more people to enjoy it. I'm from the other side. I'm a patient. I
Mhm.
mean, from a rare disease. It's not a genetic rare. Well, I think it's not a genetic rare disease. Is Cantamoeba keratitis, which is
Okay.
from the amoeba. We get it through water, and it goes through our eye.
Ah, mm,
So, long story short, I had it in 2017 and I created a foundation because a lot needs to be done because it's being misdiagnosed 80% with similarities infections. So, this sharing information and data to be able to diagnose correctly could be really a game changer for not
yeah.
only for many rare diseases. And as you said about many patients don't know what they can do with the data. That's the
Mhm.
problem. Because if many patients knew if you go after rare disease, then you say, I wish I would have known and I would have told my doctor that he's able to green light to share the information. And then maybe that is something that we need to make more noise to educate patients on the rare disease that you have the right to your data to share
Yeah.
it, to
Yeah.
help scientists like all of you to be able to do the work because we need to work together. Otherwise, we won't achieve anything, I would
Yeah.
say.
Yeah, I totally agree with you. And this is why I do my best in this field. But as a sort of data scientist, I'm with you on that, right? I want the systems to help data get shared and used. But the people who generate those data, the researchers who write the grants and create the cohorts and generate the research data sets, it's years and years and years of hard work to generate that data. It's hard to tell them just to give it away.
Yeah, no, I agree.
When the researcher in the lab next door puts as much time and effort and money in to generate a new method or a patent or a new gene discovery or a new drug, then the university tells them, don't give it away. Let's protect it. Let's set up a company. Let's all get rich, right? The one who's instead generated the data is told, oh, just give it away. So, it's competitive. Science is competitive. That's the problem. So that's the problem with researchers. They have to compete. And if they give it all away, they can't compete. And then clinicians, they don't have time. They have 10 minutes with each patient. They can't do all of this.
Yeah.
So yeah, some things need to be fixed, and I don't know how we fix them all, and it's not a technical problem. It's not a technical IT problem. It's these other things that you're talking about.
No, thank you. I agree completely.
Yeah. And I will just throw in quickly, because it's one of the things I always like to teach students. Everything's genetic to some degree. That doesn't mean to say a rare disease is an inherited genetic, single gene disorder. No. But I personally think that everything's basically genetic at the end of the day. Maybe not getting shot in the head in a burglary. Okay, some things, but pretty much everything else. If you look at the heritability of things like infidelity, that's about 70% genetic. So,
Wow.
There's a lot of genetics out there affecting how we are and who we are and what we do.
Interesting. Thank you.
Okay.
Thank you so much. And I appreciate you, Juliet, for bringing this to my attention, because I really, really care about the education. And I really think that, for example, a small task, like a small contribution to educate the patients, students, families, everyone. With almost 10,000 rare disorders have been identified. So we need to understand them. And some of them are similar. Like, for example, we have some rare cancer and we know cancer better than the rare cancer, so that is the problem. We don't have that. I'm not saying that the database is not available, because National Organization for Rare Disorder is doing a great job for collecting all this information, but that is a lot of information. We need to narrow down our thoughts and our ideas. And as Professor Brooks nicely mentioned, we have to work together, and we need to learn from each other, even including the patients in any rare disorder research projects. That's my thought. Still, I have a long way to get my PhD, but that is exactly my thought. And I agree with you 100%, and also with Professor Brooks. I have a very, very quick question. I hope it doesn't bother you, Professor Brooks. But during your presentation, you were talking about like, for example, DOC platform, and also the Beacon. So, do you think that these platforms are only working in the UK, or the database it's all around the board?
No. So the Beacon is a standard
Beacon.
from the organization called GA4GH. That's an international organization, global organization.
Yes.
So that's one of the many standards they're developing. And it's now being adopted worldwide in many different settings. The DUC-CCE is a newer standard. It only came out last summer. And we're taking a new approach there to others. So the previous approaches have been either a straightforward simple ontology, that's it, or a very, very complicated semantic model called ODRL, which, you have to have this. My brain's not big enough to comprehend or remember all the different parts of that model and how they hang together and how to use it. We've taken a middle approach where we've put a structure and an ontology together to create the DUC-CCE structure. So it's It's much more powerful than a simple ontology, but it's simple enough that people can understand it and use it. Now, the ODRL, the semantic web people say, yeah, but it can't express everything you want to express in a fully computer readable form. That's true. I never suggested it would. But now we have AI. So AI can tap into this structure in very powerful ways. That's what we're playing with now. And yeah, we've published that, and that's I know there's a project in Canada adopted it, for example. The EPND dementia platform I told you about, that's a global solution. We're putting it in there as well. So yeah, these are definitely global. They're standards. Anyone can adopt them.
That's wonderful. That's wonderful. Thank you very much. Thank you
Okay.
so much for this. Okay. Is there any more complex questions for Professor Brooks?
No, thank you very much, guys.
Yeah.
It's a pleasure to meet you.
Thank you so
We don't
much.
have any further question at the moment. So once again, thank you very much, Professor Brooks, for this engaging session, and thanks to everyone for joining us. I really appreciate that, Professor. It was fantastic.
All right. Thank you very much for saying so.
Have a
Thank
nice
you.
evening.
Thank you. Bye-bye.
Thank you. Thank you all.
Bye.